Hi, welcome to Neuroverse, a podcast hosted by Carolina
and Clara, where we discuss all matters from neuroscience to philosophy and beyond. Hi, welcome back to Neuroverse. Today's episode is about squiggly lines, an insight into learning and memory, as part of our mini-series sponsored by Scientifica, Tools for Looking into the Brain. Today we're joined with Dr. Sam Cooke, who is a group leader and a senior lecturer in neurobiology. His lab studies the fundamental cellular and circuit level mechanisms of learning and memory, as well as the causes of aberrant learning and memory in neurodevelopmental disorders and dementia. Specifically, the lab focuses on experience-dependent plasticity, how experiences and the environment shape the brain's development and function, using electrophysiological and optical techniques to record and manipulate activity in the brains of living mice in real time as they learn and form memories. Welcome to the podcast.
Hi guys, pleased to be here.
So why don't we start, as we usually do, with you telling us a bit about your research journey and how you ended up studying learning and memory in this way?
Right, well, I guess it goes back to childhood, where I would say that, one of my major interests was the natural world. So I spent a lot of time as a kid going bird watching with my dad and playing around in ponds and sort of, finding newts and stuff that I could just observe and see what was out there in the local environment, and then sort of getting very interested in more broadly the natural world, particularly animals. So I still have all those same interests that I had as a little kid. I've just put a pond into my garden. I'm still looking for newts, and so that's the start of it. But then a key thing for me to say probably is that my parents were both scientists, so that's an obvious reason that would draw me into science, and I kind of tried to avoid that initially. So that might sound like a funny thing to say, but because both of my parents were scientists and we spent a lot of time in the house talking about science, I decided I needed to strike out on my own and do something different. And so what I was interested in doing was potentially being an artist for a while. And I also used to draw all the animals that were found in the garden and etc. So I was very into that, and I went away to university. My dad managed to convince me that there's no money in being an artist. So maybe come back to that if I still really want to be a painter or something when I'm older. Come back to it. Get a university education first. So I decided to study philosophy and psychology, and I sort of managed to put myself in a corner by doing subjects at school that would not allow me to do a science career. So I did art, for instance, and media studies. So I put myself into a position where there was no coming back, which is actually something that can happen in the British education system, because you become very specialised very early. So I went to do philosophy and psychology, and over the University of Sheffield, great place to be a student. And I sort of realised across the course of 3 years that I was gravitating back towards science. Right? So I became interested in the sort of neuroscience side of psychology. I was really choosing all the modules that related to what goes on in the brain, as opposed to purely looking at behaviour. Although I still, found animal behaviour fascinating. And then in philosophy, I was also getting drawn into philosophy of mind. Even though that obviously is a massive subject that covers all sorts of things like ethics and metaphysics and all these other interesting topics. So thereafter I kind of realised that why fight it, and so neuroscience seemed like the logical next route. And particularly at this stage, I'd already really started to think about learning and memory as being something that I would be most interested in working on. I did then do a master's degree at UCL, and I worked for a brief period in that master's project on zebrafish, which is a tremendous experimental system. I should have said I had a tropical fish tank growing up full of zebrafish, so I already knew about them, hadn't realised that they were one of the major models that are used to understand development. I worked for a brief period there in Steve Wilson's lab, which was a fantastic place to be, looking at the development of axon tracts and really using zebrafish larvae as a transparent model you can look into and actually see what's going on. But there was still that draw to work on learning and memory. And so I decided to go into that, do my PhD with Professor Chris Yeo, who works on the cerebellum. So my PhD was on my first real foray into understanding the biology of learning and memory. I started to work on classical conditioning in rabbits. So this was eye blink conditioning, and it's a motor memory. So understanding how a motor memory is formed, and this sort of brought me into thinking about the different phases of memory. So we were working on not just about the learning that goes into that process, the conditioning itself, but also what happens after the conditioning to consolidate that into a memory. Okay, so really started to think about these different phases, and the psychology background was actually very useful for thinking about that as well. And from there, what I realized was we were doing invasive neuroscience, where I was actually injecting drugs to try and inactivate or inhibit certain very localized regions of cerebellar cortex and the deep nuclei, and to try and understand what was the relevant circuit to the specific response we were looking at, and more importantly, where was information stored within that consolidation window. So by inactivating the relevant regions after the animal had learned, we could exclude the possibility of a performance effect, whereby you're actually preventing the animal from performing a learned response, and therefore they're not learning fully, right? So because we were inactivating after the fact, we could say information's actually being stored here because this region is being used, activity within this region is necessary after the learning, right? And we were also able to then define a time period during which consolidation happened. So if we applied the drug within 2 hours after the learning, it would prevent the memory from becoming a long-term memory. And if we delivered it after that 2 hours, so within 5 hours afterwards, no longer would have an effect, right? So there was this brief period of a few hours after the learning, which we could call the prime consolidation window, okay? So that's all pretty interesting. But what we were not doing at that time was looking at the activity of the circuits, the neurons and the circuits that were involved, right? So I had done quite a bit of reading about plasticity and thinking about, what are the potential mechanisms by which information could be stored. Still probably the guiding doctrine in all learning and memory work is Hebbian plasticity. So this idea that individual chemical synapses can be modified in their strength to store information in a lasting and retrievable way. So then what I wanted to do next after I finished my PhD was to directly look at this plasticity. So I went to the NIMR, which was the National Institute for Medical Research, which was an MRC institute in Mill Hill, North London. But I went to work with Tim Bliss, who was a fantastic person to work with, first of all, but he was also the person that discovered long-term potentiation. So it's kind of the textbook phenomenon of an experimental model that you can use to study Hebbian synaptic plasticity, so increased strength of synapses as a result of patterned activity going into them. And what we were doing there was looking at molecular mechanisms that operate at the synapse to mediate those different phases of learning and memory. So the acquisition, the consolidation, the long-term maintenance, using relatively now old-fashioned genetic engineering technology to knock out the genes that encode the proteins and therefore not have the protein, and show that where animals lack learning and memory of specific types, particularly we were focusing on the hippocampus now, so moving from the cerebellum into the hippocampus, they also lacked the equivalent phase of plasticity, right? So at the time this was seen as relatively cutting edge, okay, and there was actually, you could look at in the literature, you'll see about a couple of decades where there was this explosion of high-profile publications based on synaptic plasticity, where someone would knock out a gene, so they'd knock out a key mechanism and then show that this also disrupted LTP, ergo LTP is the key mechanism of memory, right? We're talking like Cell, Neuron, Nature Neuroscience papers, plenty, and became this kind of formula of how you do it. And then people started to question whether this was actually the most informative approach, because it turns out that you could knock out hundreds of genes, impact memory, and also impact LTP, right? But that didn't tell you that LTP was the core mechanism. There were other interpretations always to whether it was actually necessary or even sufficient. And I should say, by the way, this is a very rambling answer to this question, but I should say that it was in Tim's lab that I first learned electrophysiology. So that's the squiggly lines. That was my first exposure to squiggly lines, you could say.
Yeah.
Although, in fact, almost every phenomenon you can represent as a squiggly line, even behavioural effects, etc. But the squiggly lines in the title refer to electrophysiology. And so what I realised was the ultimate goal for me, or at least what I thought at the time was the ultimate goal, was to record plasticity occurring as an animal learns.
Mm-hmm.
Right? So rather than take this correlative approach, actually what you'd like to be able to do is, first of all, observe plasticity happening as an animal's learning, and then intervene in that plasticity and demonstrate that prevented whatever phase of learning and memory you're interested in at the time. And this is actually incredibly hard to do, right? Because when you're doing something like inducing LTP, you are usually trying to produce a very large effect. So you absolutely bombard many, many neurons and an even larger number of synapses with high frequency activity in order to produce a very large change, right? Because what you're looking for, as in most experiments, is the biggest effect size possible. And to get that same large effect without constraining yourself in that way by actually studying a living learning animal is very tricky, because usually as we are now learning, plasticity is distributed, and it's usually sparsely distributed, right? So if you put your electrode in and try and record, you're only going to pick up tiny, you're looking for a needle in a haystack essentially. Mm-hmm. And this has been one of the big challenges. So anyway, I moved to America next. So I went to Boston, and there I worked in the laboratory of Mark Baer, who was also a fantastic boss. Such a fantastic boss that I ended up staying with him for a decade. That was such a great environment. This was at MIT, and it was within an institute as well, which was dedicated to learning and memory. So it was the PCAR Institute of Learning and Memory. So you have like 9 labs, all of them interested in learning and memory. Some people working on humans, some people working on non-human primates, some people working on flies, lots of people working on mice, but from different angles and in different systems. So what we did was, having tried to get things going in the hippocampus initially, which was what I was used to working on from my time with Tim, we realised that maybe what would be better off doing is, rather than working in this area that is highly, in people's minds is a memory centre. People think of the hippocampus as being key for episodic memory. Memory probably happens everywhere, right? And so why not look in a structure that's better understood, deals with information that's much, much less highly processed, maybe you're much closer to sensory input, and then you can constrain your experiments more tightly, and you can possibly, if you use the right type of stimuli, produce that kind of large saturating effect that might give you a chance to see plasticity as the animal learns. So we worked in primary visual cortex. These primary sensory cortical regions, of course, are relatively few synapses away from where the sensory input's coming in. And so that really constrains your experiment. You can then do clever experiments where you expose a stimulus to one eye or the other, and then you've got a pathway-specific input to the visual cortex, which is very useful when you start to think about some of the predictions of synaptic plasticity, particularly Hebbian synaptic plasticity, that you'd have input-specific effects. And so we were able to see some very large effect sizes for experience-dependent plasticity, and also deprivation-induced plasticity. So not just the effects of visual stimulation, but also the effects of removing visual stimulation. So you've got a whole suite of different forms of plasticity that you can study in that regard. And I should say that having worked with Tim, who was the person that discovered LTP, I then went to work with Mark, who was the person that discovered hippocampal long-term depression, so the reverse process, which is a weakening of synapses, which was seen at the time as potentially a sort of homeostatic mechanism, or a reversal mechanism, so a mechanism for forgetting potentially, or extinction of learned responses. And it's still not fully understood what it's for, but nonetheless it's interesting to think about these Hebbian mechanisms of plasticity, but working in opposing directions. So we could see evidence that both of those are at play when an animal learns in the neocortex. What we really got stuck into, and what I have taken away to sort of focus a lot on in my own lab, is habituation. So we'll talk more about it in a minute, but it's a form of learning that's incredibly reliable and is relevant to all species. And because it's so reliable, you can actually produce massive effect sizes without having to have complex training regimes for animals. And that means that you can have a relatively high throughput as well. So there are practical reasons. This is kind of like one of the things when I guide younger people into thinking about projects is you look for very large effect sizes, an effect that's very robust and that you can reproduce reliably, so that you can then constrain your experiments very tightly as well. So working in systems where you have the ability to minimize variables. But of course, actually the main piece of advice is work on something you're interested in, which is what I did throughout. You know, even though that sounds like a very, very long journey to start my own lab here at King's, throughout pretty much from PhD onwards is this running theme of learning, memory and plasticity.
Mm-hmm. I like the title of choice, and I just kept thinking while you were speaking that your journey is like a squiggly line.
It is a squiggly line, yeah. Yes.
Yeah, so there's a couple of things that you mentioned that I wanted to dive more into, especially for our audience who might not be familiar with these neuroscientific mechanisms. So, of course, with plasticity, there's a lot of kind of theoretical work that forms the foundation of all the research. So could you explain a little bit more Hebbian plasticity and what LTP, long-term potentiation, and depression are?
Yeah, right. So the idea that chemical synapses, so that the majority of communication between neurons is mediated by chemical transmission. It's a relatively slow process, like it's a slow solution that the brain's come up with, but about 90% of synapses are chemical. There are also faster electrical junctions, but that in itself is a fascinating discovery process that took many, many decades to unfold. For a long time, people thought that it must be direct electrical communication between neurons. And there's a very interesting sort of history of scientific discourse that led to people arguing a lot about this, but arguing in a friendly way, that these kind of dialectical forces working against each other. So a guy called Eccles, who won the Nobel Prize, amazing neurophysiologist, invented lots of methods, electrophysiology methods, had said that they must be electrical synapses, and then his own work disproved that. Okay, so this is also a key thing that we're losing a little bit. I think that we need to get back, is this idea that being wrong is really, really useful. In fact, it's more useful usually. So making mistakes, almost every hypothesis I've ever had turns out to be wrong, but that doesn't stop us moving forward, designing appropriate experiments, and it's more exciting, in fact, when you get a mystifying result than have to formulate a new hypothesis.
Yes, so many PIs have told me that. Every hypothesis I come up with is wrong.
Yeah. So maybe we're selected for not being good at coming up with the right hypothesis, but actually that tells you a little bit about the process, which is that making mistakes is really important. If we get back to this issue of chemical synapses, we now know that there's this relatively slow process of communication between neurons. But long before there was a recognition that there are chemical synapses, there was even the recognition that there are individual neurons, so cells that make up the nervous system. So you can go all the way back to Cajal to actually look at this disagreement that existed between Golgi and Cajal as to whether there's really individual neurons that are communicating via some kind of communication, it's connection. And even at this point, Cajal was able to recognize, along with some other people contemporaneously, that those connections, whatever they may be, could be a site of change that would store information. And one of the reasons that would be useful is because there are hundreds of thousands of those per neuron, right? So even just you could make the simple argument that the capacity for information storage would be much greater if those individual synapses could be modified without affecting a neighboring synapse. As compared to what you could do is change the overall excitability of a single neuron. But because you have fewer neurons than you have synapses, that obviously lowers your storage capacity.
Mm-hmm.
That's not a really good reason why the synapse is the likeliest site of change, but it is one thing to take into consideration. So that's a hypothesis that you can form. And then sort of actually a lot later, a psychologist comes along, Donald Hebb. What's amazing about what he did was that without necessarily doing a lot of invasive experiments, he's able to come up with predictions about the way the brain works based on behavior. So he's taking a psychology perspective, which is you can understand a lot about the limitations of the way the brain works based on behavior. And so he came up with this idea. Actually, he wrote most of his book around the visual system. You know, we tend to think as humans about vision because it's our primary sense. And so he kind of thought about memory within the visual domain. So most people that talk about Hebbian plasticity are actually focused on the hippocampus for the reasons that we touched on earlier, which is that it's long been seen as the seat of episodic memory, your autobiographical memory, the memory of who you are. And that, of course, is what a lot of people think of as memory, is specifically that. But memory is so many other things. It's also, as we said earlier on, skills, motor learning. Learning to do something is also a memory because information is being stored that you can retrieve. You know, so there's lots of different types of learning and memory. In popular culture, we think of memory as being that conscious memory of things that have happened to you specifically, but that's just one tiny facet of memory. So Hebb really wrote his book about how objects could be recognized using the visual system, and he thought of this idea of building representations through synaptic connections. Now, in addition to the thing that we've just talked about, which is the capacity increase that you would get from being able to modify one synapse on a neuron without changing the neighboring synapse, you also have the more important opportunity to modify the influence of one neuron, one presynaptic neuron on that postsynaptic neuron without changing the influence of all of the other inputs, right? So that becomes really useful because you can then start to segregate information flow, changing the influence of different inputs. Or if you take it a step further, which is what Hebb did, what about if the change in the synapse was dependent on the pattern of activity at that synapse? So if a presynaptic neuron was reliably causing the postsynaptic neuron to produce an action potential or be very active, so it's reliably driving that activity, why not strengthen that synapse? Because essentially what that's showing is those two neurons are probably interested in something similar in the outside world because they're both being coactive, they must be responding in the same way to some statistics of the environment, right? Mm. So let's strengthen those connections. And then what about if you actually had multiple inputs onto that same neuron that were reliably coactive, right? So probably because even if they respond to different things in the environment, those two things are happening at the same time, right? So lots of learning, let's think about classical conditioning that we touched on earlier, is an association that's being formed in the brain between two stimuli, two things that reliably happen at the same time in the same place. So they are going to be driving different populations of neurons. Maybe you've got one that's let's think of Pavlov's dogs. You've got one that's responding to the taste of the meat powder, and one that's responding to the bell being rung. One population of neurons responding separately to each. But at some point in the brain, those two populations of neurons will influence a downstream population of neurons that shares input from those two populations, right? So now you've got the opportunity to integrate information and form an association, changing the activity patterns of the postsynaptic cell so that it is reliably responsive to either of those two inputs, so that when you remove one, you've effectively stored a representation of both. So you've got the opportunity to associate inputs, and that's one of the things that Hebb recognized. So he kind of came up with this rule, really simple rule, which is that if the presynaptic neuron reliably predicts the activity of the postsynaptic neuron will strengthen that particular chemical synaptic connection, right? That turns out to be a very useful. You can come up with all sorts of interesting predictions about how then we start to form representations of the outside world inside the brain by hierarchically building up those associations and forming feature detectors. So a lot of for example, our visual system, but we could go into any sensory modality, but again, focusing on the visual system, we have feature detectors, right? So again, electrophysiology, very famously used by Hubel and Wiesel to show for the first time in primary visual cortex that there are individual neurons that respond to individual, very selective features of the environment. In this case, oriented lines. And then subsequently, other people showed that at different stages of visual processing, neurons are responsive to more and more complicated selective elements of the environment. So corners, and then shapes, and then faces or objects, right? So there's a building up of complexity there, which can all really be explained by this integration using Hebbian synaptic rules. Right, so that's Hebbian plasticity. We talked about LTP. LTP would be a model of Hebbian plasticity as Hebb originally thought of it, which was that you strengthen synapses based on the presynaptic neuron reliably influencing the postsynaptic neuron. And so Tim Bliss, who I worked with, I told you he discovered LTP in rabbit hippocampus in the dentate gyrus. And he did that with Terry Alomo, so they worked together. And what they did was just a very simple experiment using electrophysiology. So they put a very simple recording electrode into the dentate gyrus, and then they stimulated the axons, the afferent fibers that bring information into the dentate gyrus from the entorhinal cortex. And they stimulated those, and they could record a response in those postsynaptic neurons. And if they stimulated with a very high frequency, what's called a tetanus, a barrage of electrical pulses, that would actually cause the synapses to change in strength for a very long period of time. So by doing that, they were guaranteeing that those presynaptic inputs would produce a lot of activity in the postsynaptic neuron. So they were kind of mimicking, in a very artificial way, those conditions that Hebb was predicting would lead to change, right? And not just change, which had been shown previously, but lasting change, because change would have to last for a long time to be useful for learning, right? And so they were able to see that, okay, in their first experiments, this could last for hours in an anaesthetized rabbit. Now, the next step was Tim then did an experiment in awake rabbits and came back over several days. And they showed that you could get this LTP, this strengthening of synapses, would last for many, many days. And subsequently, we've had people, like Cliff Abraham over in New Zealand, doing experiments showing that you can get LTP that lasts for a year or more in a rat. Okay, so it's a long-lasting effect. Now, one of the problems with the Hebbian theory is, as beautiful as it is and as influential as it remains, it's the famous writing was in 1949 from Hebb. It's still something you hear people talk about all the time, Hebbian synaptic plasticity. Most computational models of the brain will incorporate Hebbian synaptic plasticity, and most people consider it still the most likely information storage mechanism, right? But the final proof doesn't yet exist. But there is a recognized problem with Hebbian synaptic plasticity. So what happens if I strengthen a synapse, and I have this simple rule that when a presynaptic input causes a postsynaptic neuron to be active, I strengthen that synapse? What is an inevitable consequence of that?
It will just be constantly ramping up. There's
exactly.
nothing to
Exactly, right. So you just continue to strengthen and strengthen and strengthen. Now what happens to the neighboring synapses that didn't change? Now by chance they happen to be active more often with that neuron. And so they start to also potentiate. And now you're losing a lot of the advantages that you had, which was that neighboring synapses are starting to be potentiated because they accidentally happen to be active more often with that postsynaptic neuron. And eventually you would saturate every single synapse in the brain, right? And surely cause epilepsy long before that. So you've got a problem, you've got this kind of runaway train. And so people have been studying now for many decades, or particularly modelers thinking about how you can introduce homeostatic mechanisms that would maintain overall network function and keep the benefits of individual synapses being modified without bleeding over to neighbouring synapses. And so one of their first candidates was the reverse process, which is long-term depression, right? So it's actually paradoxically being called, some people call it anti-Hebbian plasticity, some people would still call it Hebbian plasticity. The idea is that if the presynaptic neuron reliably fails to cause the postsynaptic neuron to fire, then you weaken the synapse. You don't just leave it static, you actually weaken it. So now you've got bidirectional change as an opportunity, right? So that can introduce a balancing force and also increase signal to noise. So as you strengthen some synapses to represent the statistics of the environment, you're also increasing that representation by improving signal to noise by weakening synapses that explicitly are not activated by that. So there's dual benefits potentially to this or reverse process of weakening, and that's modelled through, as Mark did in the hippocampus, long-term depression, right? So there what they were doing was actually using a very strange low-frequency stimulation of presynaptic inputs, which kind of mimicked the fact that there was an action potential in the postsynaptic neuron before the incoming pulse. And then, more interestingly, people have studied a phenomenon called spike timing dependent plasticity, which you'll hear a lot about, which is just another way of experimentally inducing Hebbian plasticity bidirectionally. But here what you do is use more sophisticated electrophysiology to actually govern the activity of the postsynaptic cell artificially by injecting electrical current into it and making it produce action potentials and timing that relative to the pulse of electricity that you put into the presynaptic inputs, right? So you can actually govern the timing to have the presynaptic input active before the postsynaptic neuron at a given timing interval, or the reverse, which is that the postsynaptic neuron is active before the pre. And hey presto, you get potentiation when the pre comes before the post, and you get weakening LTD-like effects when you activate the postsynaptic neuron just before the presynaptic input.
Yeah.
So that's again a very long-winded answer to your question.
Yeah, I like the emphasis on the usefulness of electrophysiology, particularly to study plasticity, because it's never been so clear to me before of plasticity of most of the mechanisms that happen in the brain in particular really is activity dependent, and so it requires, reading out and manipulating that activity to induce it or to observe it. And that's a really good demonstration of the use of electrophysiological tools.
Yeah, I think it's still the go-to set of techniques that people use to study the brain, because the brain is actually electrically active, right? So studying...
Even though the synapses are chemical.
The synapses are chemical, yeah. But
Yeah.
the things that you're recording from the neurons are electrically active. As you know, and I think you've had other podcasts talking about this, there's now a big push towards optical imaging, and also interventions that use light, and those are really sort of revolutionizing neuroscience experiments. You can that kind of spike timing effect, you can also produce that with optical techniques, because you now have the ability to sort of image the activity of a neuron at the same time as stimulating it. And so potentially in the future we will move away from electrophysiology, but at the moment the tools are not quite there in terms of temporal resolution, right? So while there are many advantages to imaging over electrophysiology, for instance, you can look at hundreds of cells at once, whereas with electrophysiology it's very, very hard to do that. With electrophysiology you're measuring in real time with high fidelity what's occurring in a neuron.
Going back to learning and memory, I really enjoyed your explanation of Hebbian plasticity and how it allows you to have an integration of information and essentially an internal model of the world.
Yep.
And I wanted to ask you about what goes wrong when the internal model is disrupted. So when prediction error occurs And I'd like to bring in this paper that you have about electrophysiological signatures of visual recognition memory across all layers,
Right.
which I found really interesting, in that you spoke about this mismatch activity, and you had a behavioral paradigm where you gave mice familiar images and then you reversed these images. So could you explain a little bit about this behavioral paradigm and what you mean by mismatch activity and how that affects learning?
So habituation's become a big focus for us, and that actually captures lots of processes, right? Because habituation can occur over different timescales. People have different definitions about what habituation is. So we regard habituation as essentially learning to ignore anything that's useless, right? And so you might say, well, why study how we learn to ignore things that are useless? Isn't it more important to study the things, how we learn about what's important, what's rewarding, what's punishing, etc.? And the answer to that is that you can't do that unless you can determine when something's useless, because your brain is overwhelmed by the amount of information that's being presented to you. So actually, from the moment you're born, you're habituating to your environment. And in fact, if you look at babies, one of the only forms of learning you can really study is habituation, because they can't talk to you, right? So, and that also goes for animals. It's relatively easy to study habituation because they all do it. Even the simplest animals habituate, because essentially their task is to find the things that are rewarding and to avoid the things that might punish them. And as a necessary consequence of that, they have to explore things that are novel. Right? So that's all species are interested in novelty as well. And that's because that novel element of their environment deserves that exploration, because it may be punishing or it may be rewarding. So you've got to establish that. But once you've established that it's not rewarding or punishing, it becomes familiar but useless. And so it's really important that you don't expend energy or particularly attention on that stimulus, and you devote your energy and attention to things that are novel, things that punish, and things that reward. So it's a very interesting process that there are multiple solutions to it, because you see, organisms that have incredibly simple nervous systems habituate, all the way up to us, where you could argue that the majority of our learning is actually habituation. When you go to a new environment, you are a little bit overwhelmed with it. It is definitely going to impair your ability to carry out the tasks of the day, but you get better at it as you start to get used to the environment, and then you can focus on what you need to do. And so you can then do all sorts of even more interesting things, like plan what you need to do in the day, or solve problems, because you are not actually responding to the sensory environment. But the brain has an additional interesting problem, right? So one solution to that might seem very simple is, well, I'll just weaken the response of all neurons to that. Maybe I'll just have LTD. I'll LTD all the synapses that are responsive to all the stuff that's not rewarding or punishing or I've not seen before. And that's a simple solution to habituation, or ignoring those things, because they won't then impose themselves on the activity of the brain. But you've got a little bit of a problem in that when something changes about that stimulus that might become useful in a different context. Let's say you're in a meeting, you're ignoring a conversation that's going on next to you, but suddenly your name gets mentioned. You can immediately dip into that conversation, right? So somehow you have the capacity to monitor everything that's going on around you. So you're still monitoring the environment, that all of that stuff that you're ignoring is still having an influence over your brain, but you're able to not dedicate attention and energy to it. So that's rather fascinating, right? That suggests it's not, in some cases it might be, in some cases it might be actually weakening of synapses, but that can't be the whole explanation. Something else has got to be going on. So there are multiple different theories about that we're working on. So the paper that you're referring to, something that we've been looking at for a while, which is a very long-term form of habituation, where if you expose the animals to a very simple visual stimulus for, let's say, half an hour, you find that they're initially highly responsive to it, And actually, if the animal's moving around freely, it will go over to that stimulus and it'll explore it, because, as I said, it's a novel stimulus and the animal needs to explore it to find out whether it's useful. It could be a really abstract stimulus. What we usually use are just lines, right? So it shouldn't, there's nothing intrinsically interesting about the stimulus, but because it's new, the animal explores it. You can also just, have the animal sit still, and the amount of movement that it produces indicates that it's noticing the stimulus and it's responding to it. If we come back the next day, then the animal will respond much less to that stimulus. So there's something going on in between times, and we talked about consolidation. There's something going on in between times which is changing the brain so that it's no longer responsive to that. Interestingly, this particular form of plasticity requires sleep, right? So if the animal doesn't sleep, it doesn't happen, right? So there's a long-term change. Now, if we just look within the session, the first time the animal looks at that stimulus, it will gradually reduce its exploration throughout. Let's say you've got 20 minutes, half an hour, it will reduce its exploration of that stimulus. So you're seeing habituation in real time happen. And then when you come back the next day, there's actually some savings from one session to the next. Interestingly, actually, very young animals, babies, for instance, don't show the savings. So you can show them the same stimulus again the next day, and they're just as alerted to it and interested in it. So that kind of the long-term savings, whatever's going on there, doesn't start to happen until you're at 6 months of age.
That really demonstrates how habituation is memory, because it's like, if you were to tell me that in the non-habituation context, I would just be like, oh, babies can't remember things.
Yeah.
But you could also think of it as like pure habituation in a way where it's just like, oh, they see more things as novel, but then it's the same thing.
Yeah.
Yeah.
Yeah, I mean, there's lots of interesting things related to the age, right? So more things are novel, as you say.
Yeah.
Right, so you're constantly overwhelmed by your environment when you're young, but you're also exploring everything, right?
Yeah.
And then as you get older, things become a bit more blah, because you get used to everything. And you could call it wisdom, I suppose, but at some point there's nothing that surprises you.
Yeah.
And it's just quite interesting to think about the aging process, right? Because in addition to that, we tend to, as we get older, seek familiarity. So when you're young, you're seeking novelty a lot. There's an additional thing that's happening that probably relates to the reward of the novelty itself, whereby you're going through this transition in life, and that you go from becoming a novelty seeker to a familiarity seeker. But maybe seeking familiarity, putting yourself in a position where, I've got a house now, I like my house, I like to hang out in my house, I like my routine, I like having coffee at the same time every day, I like taking the same route to work. You're limiting, in some ways, your experience of the world. So that's kind of interesting to think about how, we think of this aging process and cognition as changing radically as you age, but a lot of that might be guided by our behaviour. There might be an opportunity for older people to force themselves to do new things and reinvigorate all of those processes that lead to learning. The other thing relating to that is our perception of time, right? So when you're young, you think about how long things take very differently from when you're old, right? Things go past in a flash as you get older. Suddenly, like, months go flying by, and you can't even remember what happened in those months. Whereas when you're young, it's like a car journey. It can seem like it takes forever, right?
Maybe because everything is so much more salient, so there is generally more.
Well, yeah, exactly. But partly because so you're forming new memory when you see something novel. And as you start to see fewer and fewer things that are novel, actually the unit of time is really memory, right? So you've got fewer of those units as you get older, and so just time is different. Yeah. It's memory. Time is memory, really.
Yeah, and with habituation, I guess the days just blur together, and so as you get older, you get fewer novel stimuli, then you get fewer of those spikes, like attention spikes. Yeah, exactly. So it makes sense that time kind of like compresses as you get older.
Yes, but maybe we can find ways to stop time compressing by learning to tango or whatever.
So to link habituation back to neural activity, going back to what you were saying about showing these mice these simple visual stimuli. So you mentioned how they habituate in terms of behavior, but can you explain how this relates to squiggly lines?
Squiggly lines, yeah. So, well, one thing we can do is turn behavior into a squiggly line. So we do that. So you can actually, as I said, turn anything into a voltage signal that you can then measure. And it doesn't have to be the electrical activity of a neuron or neurons. But this is the key that you're asking this, right? So what happens in the brain? So we use electrophysiology to record the electrical activity, sometimes of individual neurons. Sometimes we might record the spiking output of neurons. Sometimes we might record the synaptic input, as in the LTP experiments. And in those cases, you can either give yourself a really hard experiment where you try to record intracellularly, the kind of stuff that you're doing, Clara, where you're actually, recording inside the neuron, and you're possibly, in the best case scenario, able to record individual synaptic events. But another way you can do it is to record the population activity of lots of synapses, and that's what was done in the original LTP experiments, where because you're stimulating lots of inputs, you can actually record a population level signal that's so large, even though each individual synapse produces a very small potential, you can, if you've got hundreds of thousands of them being active at the same time, that will produce an effect that you can record even with the electrode not being inside the cell but being outside the cell. So this is a signal that's called the local field potential, and it's actually very similar to a slightly even larger scale signal, which is the electroencephalogram, so the EEG signal that people record in humans just by putting electrodes on the scalp, right? So if you've got enough synapses being activated at once, you can record a population level signal. And even though synaptic events are much smaller than action potentials, the output of the neuron, they actually summate a lot more because they're slower. We talked about chemical transmission being slow.
Yeah.
Well, they're actually very slow, and because they're slow and there's a lag in them, even though they're happening at slightly different times, they summate together to produce a very large signal, whereas action potentials happen really, really quickly. So there's much less opportunity for them if there's at slightly different times to summate to produce as large a signal. So paradoxically, the smaller synaptic signal is the one that you can more readily record at a distance, because it can become large enough when enough synapses are operating more or less synchronously. And so that's why the EEG is mostly a synaptic signal. So anyway, these are the squiggly lines of which we've been talking, and what we do is use all sorts of different recordings. So we record action potentials, but we're also recording these local field potentials recorded within neocortex, so recorded in that primary visual cortex as the animal's viewing visual stimuli. And if we record during that first session, which is when the animal's exploring the object, is highly engaged, and then, or in this case, just a two-dimensional stimulus, and it gradually explores less and less, what we see is a reduction in that cortical response. And that's kind of exactly what you'd predict if we have our, why not just weaken all the synapses and reduce the influence of that stimulus on the brain, right? So that if you've got that hypothesis that you're what you might call adaptively filtering out that stimulus, you just weaken the synaptic connections that mediate response to that stimulus. However, really interestingly, if we come back the next day, we're recording through the same electrodes in the same position, mostly the same population of synapses, the response has doubled in size, right? So whatever happened during sleep has gone from the cortex weakening its response to that stimulus to now dramatically increasing its response to that stimulus. So what's going on here? Totally weird. Those are the most interesting things to study often, are the things that don't make a lot of sense initially. And here's where we're getting into thinking about a more complex circuit that's going on here, which involves not just excitation but also inhibition, right? So you could also imagine a system in which inhibitory neurons are involved, so GABAergic neurons that are using a different chemical at synapses to suppress the activity of the postsynaptic neuron, right? So another thing you could imagine happening in habituation is selective inhibition is happening, so you're actually suppressing the activity of those postsynaptic cells through inhibition. That also is not full explanation of what we're seeing, because what we're seeing is an increase in the activity of that region, right? And what we're starting to find is that there's some very complex things going on where different types of inhibitory neurons are interacting, and they're basically serving as a switch between memory and experience.
I just wanted to ask a question about two specific things you mentioned. So the first one being timescales and the second one being interneurons or the mechanisms. As you said, there's different mechanisms that seem to be happening at different timescales. So, sleep-dependent mechanisms seem to be different from the acute adaptation that's happening as soon as the stimulus is repetitively shown. And do you think that the kind of key behind these different mechanisms is the involvement of different cell types?
Yes. There's actually more timescales than I've mentioned to you, right? So actually we have things happening over milliseconds, we have things that are happening over seconds, we have things happening over minutes, we have things happening over days and days. So there's lots and lots of things going on there. And, the brain as an evolved system has many different solutions to problems, and they probably are operating in parallel all the time. But yes, we certainly have some indication that, interestingly, some of the same inhibitory neurons are involved in both that process that's happening over the minutes of the session that you're working on versus from day 1 to day 2, etc. But they're not involved in the same way. So very popular in neuroscience at the moment is this idea of looking at circuits, canonical circuits, or mechanisms that seem to be almost engineered to deal with a certain thing, like a comparator. One might be a comparator system. So a comparator system is a way of comparing memory with reality, right? So to detect novelty, what you ideally have is a comparator which says, have I seen what I'm looking at? So you impose your memory on the environment effectively, which gets into all these sort of ideas that you've probably talked about before, like predictive coding, right? So you're imposing your memory on the environment all the time, and if you get a violation of that model or prediction, then you engage in learning. So you have to have circuits in the brain that serve as a comparator. And one nice way to do that would be with inhibition that perfectly matches excitation, right? So if your memory is imposing through inhibition equal strength of influence on a neuron to the sensory input that's coming in, you get a net zero output. So that's a comparator circuit. If you change either one slightly, if there's no match, now you produce activity, and so you get this kind of violation, and therefore, hey presto, your brain engages, right? So for example, one thing we're working on at the moment is, if we take one type of inhibitory neuron, which are these somatostatin, a protein that is widely expressed but is particularly strongly expressed in that type of neuron. The key thing here is that you're able to find cells that are inhibitors of inputs. So I kind of think of things slightly crudely as where inhibition is applied in the dendrites, where synapses are on excitatory cells, you're effectively applying inhibition to the input to that neuron. Where another type of inhibitory neuron, like a fast spiking inhibitory neuron, a basket cell or a chandelier cell, actually applies all of its inhibition to the output of the neuron. Right, so it's actually stopping, it's shunting inhibition, which is stopping that neuron from producing action potentials, even though everything else is going on as normal. All of the other flow seems to be normal, right? So somatostatin expressing inhibitory neurons are applying their inhibition in the dendrites, but they're also doing something else interesting, as it turns out, which is that they're also inhibiting those fast spiking inhibitory neurons. So you've got two things going on. Direct inhibition, those same neurons both have the capacity to inhibit their excitatory targets at the same time as applying inhibition to another class of inhibitory neuron, and therefore disinhibiting their excitatory targets. So what we think is going on maybe is those same neurons are involved in different timescales, but they're involved in different ways. Maybe in one case applying inhibition, and in the other primarily acting through disinhibition. How that happens, how you get those selective modifications, potentially what's happening is that the inhibition to inhibition synapse is the area that's being affected during sleep, for instance.
I'm glad you went into that level of detail, because that's kind of what I was trying to get at. That subcellular specificity of the inhibitory control is so interesting to me.
Yeah.
And it just adds like another level of computation, essentially, as you can control whether the inputs are being changed or the outputs are being changed of the single neuron.
Yeah, so that's something that requires a lot more work to understand. At King's here we have lots of people that work on these different classes of inhibitory neurons and the logic of their connectivity. So a lot of work across the world has been done on this, characterizing these different types of GABAergic neurons and the fact that some of them largely only inhibit GABAergic neurons.
Yeah.
Some of them very rarely inhibit other subclasses. Some have this mixed effect, And so you can imagine that you get inhibition, you get disinhibition, and you also get disinhibition.
Yeah.
And God knows you probably get even more complicated things happen. And then the other thing to think about is this top-down effect. So we've just talked about this idea of memory imposing itself on perception. Well, how does it do that? It does it by the whole of the brain having the opportunity to feed into primary sensory regions and influence that. So that's that top-down effect. And maybe one of the routes in is through these inhibitory neurons that could potentially serve as comparator systems. But there are different routes in, right? So there might be a different influence of one type of top-down effect from another coming in through different subclasses of inhibitory neuron. All as yet not well understood.
So much to disentangle.
As I said to you, your brain still has to respond to the environment even when it feels like it's fully habituated to it, because something can happen in that environment that you suddenly need to attend to. So what we seem to be going into is a gating system where sensory input is having the major influence over visual cortex when you're actually learning and attending to it. And then memory is imposing itself once you've got an expectation or a prediction of what's going to happen, where you're habituated and you know that that's not that useful. But there has to be something else that's an alerting signal that can happen, that can break through that suppressing influence of memory to make the brain responsive. And because we're using dynamic stimuli, right? So because the stimulus is moving, or in this case it's flipping, you actually have this dynamic stimulus and it's causing a bursting activity of neurons. So while those neurons are suppressed by inhibition, there's also something complicated going on which allows really strong stimulation, moving stimuli, to break through that inhibition and alert the brain that something's changed. Right, so that's, in its most extreme case, you could think about sleep, right? So when you're asleep, you think that you're locked away from your environment. But actually that's not the case. So if you're a parent that has a young child, that child crying in a different room on the other side of the house can wake you up. Right, so what is it that allows that sensory stimulus to penetrate your brain and wake you up when equally loud stimuli, like an ambulance going by, doesn't break through? So somehow your brain is still paying attention to the environment and selecting out the stimuli that will wake it up and managing to ignore all the stimuli that are not that useful. So
wait, going back to the increased activity, what do you hypothesize that is? Do you think that that's potentially the underlying attention mechanism that then you can act on? Because from what I understand, when you are exposed to the stimuli, that's the sensory experience, and over time you habituate to it, which is why there's a decrease in activity during the session. And then the next day there's an increase in activity. So is that increase in activity purely just the memory? So I find it odd if it's expectation, because it's not violating the expectation, right? Unless it's potentially confirming the expectation.
So no, you make a good point. So this interestingly relates to the methods that we're using as well. So I've told you that we use these local field potentials. So one commonly used method that we use is that because we've got these dynamic stimuli, we've actually got what's called a phase reversal, where if you imagine a black and white striped image, all of the white flips to black and all of the black flips to white, right? So and if that phase reverses, or it's dynamic, it changes in that way systematically, very regularly, then you can actually drive activity. So you can what's called evoke a response in the nervous system. So if we're recording in visual cortex, you can evoke a thing called a visual evoke potential. And this is one of the most commonly things measured even with EEG, right? So the size of that potential, people tend to talk about the magnitude of the response as being reflecting the amount of activity that's going on in the brain. Now the paper that you just sort of mentioned, that was one of our recent papers, is very interesting because what it reveals is that it's not as simple as that. So it's actually relevant not just to our own interests of habituation, but also thinking about methods, right? Because if we record the spiking activity, i.e. the output of neurons, yes, when the stimulus changes, when it's phase reversing, you're getting an increased rate of action potentials, right? So something is happening just as the stimulus changes to produce an increase in the activity. And I know that I'm waving my hand here in imaginary squiggly lines, which you can't see through the podcast. But I'd like you to, imagine squiggly lines, right? So you get this peak directly after the stimulus in the amount of action potentials. And that lines up very nicely with this larger visual evoked potential that's produced at the same time, because there are synapses that are synchronously active because of that change in the stimulus, which are driving that spiking activity. Right? So those two signals seem to line up really well. But then when you look at the spiking activity of the neurons, what you see is that they then dip to almost nothing when the stimulus is familiar.
Yeah.
But if the stimulus is novel, they fire at a much higher rate. So in fact, it's not true to say that the cortical response is increasing, because if you look throughout the whole period of stimulation, there are actually, if anything, more action potentials produced by the novel stimulus than the familiar. However, when the stimulus changes, that instance when it changes drives more spikes for the familiar than for the novel. So that phasic response is bigger, but the continuing response is bigger for the novel.
Why do you think this phasing response is larger for the familiar?
So I believe this is something that we're working on. Different sources of inhibition are combining to create a situation in which there's some kind of bursting activity that's driven by a dynamic stimulus, whereas when the stimulus is static and you've habituated to it, there's no longer any spiking. So one hypothesis, which we're a long way from being able to test, but we're thinking about how you would test, is that this is a change detection system. So I've told you that you still have to have the ability to monitor your environment even though you think that you're fully habituated to it. And again, if you think about the baby crying, something happens in your environment that breaks through ignoring your environment to tell you that something's changed, right? In this case, we're fooling the brain by just having this dynamic stimulus. And actually, it's interesting that moving stimuli are somewhat hard to habituate to.
Yeah.
And it's in a sense, you can see people get mesmerized by screens with moving images on them. They can't stop looking at them, right? Because they're artificially dynamic. There's always action happening, right? And in our environment, particularly, our offices, there's not a lot of dynamic stuff. So that's an interesting thing to think about. I don't want to come down hard on explaining exactly how it happens, because we're talking about being right at the sort of, this is exactly what we're thinking about and working on at the moment, the great unknown.
Yeah.
But one thing that we can take away is that the visual evoked potential is not exactly always what we thought it was, right? It's not always bigger means more.
I guess that's also an important point, that the benefit of doing this with the mouse work is you can be more invasive and understand really the mechanisms that are going on, like complementing it with optical techniques to manipulate these interneurons and whatnot, and then you can translate that to whatever evoked potentials you're recording in humans, maybe with EEG, that we really don't know what that really means, and it's mainly correlational in terms of neural activity with behavior. And it's useful to have that link with the LFP recordings, where you can dive a bit deeper into what these signals are actually reading out. So
that brings us to thinking about translation, right? So we talked a lot about fundamental work, and so the career trajectory that I told you about earlier is nearly all fundamental exploration of brain function, not thinking about disorders or altered learning and memory, but thinking about typical brain function, okay? So what about, again, as you're saying, thinking about how we can translate, because I truly believe, actually, that I can go off in a big digression about this, but I truly believe that working on the way things work as they normally are is the best way to understand what goes wrong when there are changes in the brain. But we do have to be able to translate our fundamental understanding of the brain into thinking about human conditions that need treatment, etc. And one of the aspects of that is thinking about how you model a human disease in an animal. That's been a very complex process. It's always challenging. The other thing that you can look at is just simply those measurements that you're talking about, right? So if we think about EEG, it is something that is widely used. Turns out EEG, even though it was discovered, I guess a century ago, is actually still in use as much, if not more than ever. And it's a very crude signal that can be recorded cheaply and easily. You can even record EEG in your own home. So that's becoming more and more of a thing, as sending people home with a little piece of equipment to monitor their own EEG through sleep, etc. You can't do that with some of the more sophisticated techniques that have been developed in recent years, like imaging techniques, functional magnetic resonance imaging, etc. Those still require very large, expensive pieces of equipment. But EEG is cheap, so maybe we can still make use of it. And indeed, people are kind of cottoning onto this fact that as there's more and more financial pressure on healthcare systems, actually EEG is a very, very useful thing to measure. But the problem with that, and this is also true, by the way, for the more expensive things like fMRI, is that we don't understand what they actually are measuring. Mm-hmm. Right? We know that fMRI measures oxygenated blood flow, but we don't know what different patterns of activity produce in terms of oxygenated blood flow. So what are you actually looking at in terms of neural activity? The same is true for EEG. When you record EEG, you record a signal like a visual evoked potential, or maybe you record oscillations in that EEG. So, different frequencies of waves, again, going back to our squiggly lines, waves that are occurring at different frequencies, which we think indicate different circuits and different cell types being engaged in the brain. But we don't really have that proof. So the only way to really understand that is, as you say, to go into a system like a mouse. First of all, maybe record a bit more invasively, so go a bit closer to where the action is happening, like with our LFP experiments, but maybe also intervening, stimulating a particular type of neuron and seeing what the consequence is for the EEG signal. Then you gain a bit more proof as to what that EEG signal means. Mm-hmm. Knocking out a receptor. We think of lots of drugs that are used in psychiatry at the moment. Let's take something like psilocybin. They don't really know fully how they work. They think that they work on 5-HT2A receptors, right? But there's probably a little bit of bleed over into other receptor systems as well. But they don't know which neurons it's binding to, right? Mm-hmm. So you could genetically knock out that receptor in just one type of neuron and not others, mm-hmm. and see whether the drug is efficacious, see how that affects the EEG. So you've got lots of different angles in once you start to do what's called reverse translation. But if you think about the things that are important to clinicians, for example, neurologists, psychiatrists, etc., you think about the things they're actually interested in doing, either in terms of what they measure or the substances that they're applying. They don't necessarily know how they work, they just know that they do work a little bit. They don't work optimally usually, but they work a little bit for treating depression, for treating schizophrenia. Can you work out what those medicines do? And by working out the underlying mechanism, then iteratively choose another substance that works even better, mm-hmm. right? So there's a whole process there that we need to start engaging in, which is finding out what clinicians and other people that work with patients are interested in, both in terms of measures and treatments, and then using our model systems, whether they be animal systems or they be cell systems or computational models, to dig into those. And at the moment there's that big gap, people call it like the valley of death, right? Mm-hmm. Which is this gap in between fundamental scientific research and translation, whether mm-hmm. that's into healthcare or technology development or whatever, right? Maybe it's easier to get back across the valley of death if you start over where you want to get. Mm-hmm. And in fact, it will be an iterative process back and forth, but it's a change of viewpoint, because I think for a long time we've been focused on what's called forward translation, which is like trying to make a mouse into an autistic mouse, for mm-hmm. example. We all know that there are challenges there that are difficult to surmount.
Yeah. Really interesting, and I especially love now that I've learned this new idea about reverse translation, and hopefully that will help inform fundamental science as well, and that there'll be a feedback loop. So once we understand a bit more on what's happening on the translational side of things, we can then also better inform our fundamental research.
Yeah, I think it's the great challenge for those that sort of fund science. It's been the great challenge to get those two schools of people working together. You know, there's always been a great push to have clinicians actually go into the labs. I can think about people in neuroscience that were clinicians that then went and did very, very fundamental work. So they're actually doing that reverse translation by training themselves up to be experts on disorders of the brain, and they then go and work on something very fundamental.
Yeah.
So you can think about someone like Eric Kandel, for example, who, you know, did all this amazing, very fundamental work in aplysia, sea slugs, you know, on synaptic plasticity, but he was a psychiatrist, right? And then we've even got Carl Dieseroth, right, who's one of the key inventors of optogenetics, that technique that we're all using, right? But he also is a psychiatrist. Right? So because he's got that perspective on what's needed, if he himself ventures into the lab and starts engaging in really fundamental stuff, he's bringing a lot of knowledge to the table. But yeah, we could list lots and lots of people. But I think looking at the discovery process is really important as well. So I told you before about my views on the importance of fundamental neuroscience. And if you look at the discovery process, so many things are discovered serendipitously.
Yeah.
It's very, very rare that you find what you're looking for. All right, and so scientific journeys take you in all sorts of directions. What's important is to be able to go with the flow a little bit and go where it takes you. And, you know, think about all the crazy sort of things that people have done that have transformed the serendipitous elements of this. Like I always talk about when I'm teaching students about why Hodgkin and Huxley, you know, knew about feedback amplifiers. So the ability to actually control the voltage across the membrane of a neuron by injecting current into the neuron. A lot of that knowledge and expertise only came about because they had been in the Second World War working with radar technology, right? So that they're actually bringing something completely outside of what they work on into neuroscience, and that transforms neuroscience. And then they, of course, have to work on a neuron that's large enough that they could, at that time, using the technology they had, get a probe into the neuron to inject the current in. So they have to work on a squid giant axon. And so they, you know, that's where all of these breakthroughs come from, is necessity, but also serendipity. So I think really teaching people about the discovery process can make people aware of the importance of really, really fundamental stuff.
Yeah.
I think that's a great place to wrap up the episode. And yeah, I feel like we've been on quite a journey this episode. Thank you so much for going through all of the theory behind the plasticity and then also expanding and relating to your own life. It was a really nice journey.
Well, thank you very much for having me. I've enjoyed rambling, I would say. It's somewhat of a ramble through things, and yeah, thank you very much.
Thank you. It's been very informative and super interesting. Looking forward to future papers that come out of your lab.
Thank you very much.
Thank you so much for listening to this episode. It is part of our series sponsored by Scientifica, who are one of the leading producers of state-of-the-art electrophysiology and imaging tools. Scientifica supports researchers around the world, making leading equipment, but also hosting an extensive resource center. So be sure to check out their website for more information if you're interested in these sorts of tools. And if you enjoyed this episode, please share it with your family and friends, and give us a follow on Instagram and Twitter at neuroverse_pod.